Good morning, dear colleagues. First of all, I would like to thank for the invitation to make a talk on the Chinese-Russian workshop. And my topic is multi-parameter optical diagnostics of microcirculatory tissue systems, methods and technical means. And I would like to introduce you to our results of research on this topic. Just a little bit information about where I'm from. A little more than 1,000 kilometers separates us from Saratov now, unfortunately for all of us. Here is information about our university. Now our university, named as a flagship university, has become the largest university in our region, and I will show you some results with implementation optical non-invasive diagnostics to real clinical practice. Here is more detail presents the main direction of our activities in various fields of medicine, and such as endocrinology, rheumatology, dermatology, minimally invasive surgery. It's important for modern medicine to have non-invasive, fast, reliable, and comfortable diagnostics. My talk will include the following sections: a little bit information about our object of study, microcirculatory tissue systems and diseases associated with conditions, and information about multi-parameter approach in application OND for study microcirculatory tissue systems, and I will show you some examples with our multi-parameter optical non-invasive diagnostics application for some clinical practice. For example, it will be functional diagnostics, rheumatology, endocrinology, minimally invasive abdominal surgery, and our new results in neurology. Of course, and the end will be conclusions and acknowledgements. Just a little bit information about object of our study is a very complex physiological system, microcirculatory tissue system, which include not only arterials, capillaries, etc. Microcirculatory tissue system is the main structural and functional unit of organs, providing a trophic adaptation, regulation of the individual microregions. Sorry. And at this slide, we can see diseases associated with the conditions of microcirculatory tissue systems, blood microcirculation and metabolic activity of human tissues investigated in our studies for diabetes, functional diagnostics, rheumatic disease, and minimally invasive surgery, and so on. Our approach is here. The proposed multi-parameter approach is based on the following relationships between the parameters of microcirculatory tissue systems, such as perfusion, peripheral blood flow oscillations, arterial saturation, tissue saturations, and so on. And microcirculatory systems conditions in various diseases. For example, the five main these conditions such as functional changes of capillaries, endothelial dysfunction, venous congestion, ischemia, arterial hyperemia, and with possibilities of optical non-invasive diagnostics methods. Thus, the combination of several complementary O&D methods with the simultaneous performance of functional tests provides more valuable and reproducible diagnostic information for assessing the functional state of microcirculatory tissue systems. Our multimodal, multi-parameter approach, we using very well-known technologies, but our main goal is to raise them to the level of wider application directly in the clinical practice of a doctor. We use I will be showing you examples with such very well-known technologies, laser Doppler flowmetry and video capillaroscopy, diffuse reflectance spectroscopy, and fluorescence spectroscopy. This is our main idea. And new application approach of O&D for medical practice for functional diagnostics. To prove an answer for the question whether the LDF records were really proportional to red blood cells velocity, we have prepared a laser Doppler monitor and high-speed video capillaroscopy. This is our experimental setup. As you can see, video capillaroscopy methods allow to register and quantify such features as reverse blood flow during occlusion test. I mean, here you can see. And it's very interesting these results for two methods, video capillaroscopy and laser Doppler flowmetry. The curves reflect a high degree of correspondence between the local variation of blood perfusion and blood flow velocity before and after occlusion. We can see here. And by evaluation of wavelength coherence spectra, we have come to conclusion that this oscillation in pulse band, you can see here, is around 1 Hertz. Pulse band highly correlates with the same oscillations in a single capillary. This is very important. The laser Doppler flowmetry techniques can be easily combined with different types of measurements or to significantly increase specificity and sensitivity of diagnostics. The perspective direction of development of optical non-invasive diagnostics methods is an extension of a technology in the field of mobile health. This is LDF and fluorescence spectroscopy and tissue reflectance oximetry system, and this is now. It's something like a gadget, a fitness bracelet. At the moment, the Russian manufacturer of LDF device Lazma has already released the first samples of wearable devices. We conducted a pilot studies to evaluate the synchronization of LDF signals, taken at identical points of the different hands. In our study, to study the synchronization of blood perfusion in the contralateral limbs. A wavelet coherence analysis was used. Just a couple examples for rheumatology. It's a big problem in medicine, microcirculation disorders. I mean, microcirculation disorders in the end lead to hypoxia and tissue edema, the appearance of neurobiotic processes. And here is shown to you combined methods to assess microcirculatory disorders inflammatory activities. In patients with rheumatic diseases, we produced a new protocol for study. Here is our experimental setup. Here is a typical LDF grams and curve with cutaneous of thermometry, skin temperature, and we proposed new diagnostic criteria from two methods. And again, in the end, we proposed a new a method for clinical practice. A new version of this study was using together laser Doppler flowmetry and tissue reflectance oximetry. Again, we produce a new protocol, and in the end, we obtained results with a high sensitivity and specificity. And this is important for real clinical practice. And next our study for rheumatology it was with combine two methods again, laser Doppler flowmetry and video capillaroscopy, image of a capillary bed for conditional healthy in the left and for rheumatic diseases. Here, again, a new protocol. And at this slide, we can see a different reaction to a cold pressure test. And very important to analyze a velocity of red blood cells and LDF signal, because for for patients with rheumatic diseases, this reaction is different. It was our pilot study. For endocrinology, again, a big problem, as well known. Again, we try to combine two technologies in this case, laser Doppler flowmetry, fluorescence spectroscopy, and diffuse reflectance spectroscopy. And we obtained these results, a different reaction for temperature tests, heating test for healthy volunteers and for patients with diabetes. And in oscillations, we obtained different results too, especially in myogenic and respiratory ranges. And again, we obtained with high sensitivity and specificity results. And our fresh result for minimally invasive abdominal surgery, as well known, this a big problem in medicine, in minimally invasive surgery, because it's important for a surgeon to know the oncological status of the tissue viability, the presence of blood flow, and so on. Of course, now using a lot of optical diagnostic techniques, technology, as fluorescence spectroscopy, and again, our multi-parameter approach is to combine fluorescence spectroscopy, LDF, and diffuse reflectance spectroscopy in one optical fiber. Here is our multimodal experimental setup with two methods, our experimental study were conducted at the Department of Interventional Radiology of Oral Regional Clinical Hospital, and we try to study a common bile duct, gallbladder, and liver abscesses. And our results for fluorescence is here for excitation ultraviolet and blue wavelengths, and for microcirculation, atypical is shown here. And again, we obtained a different, significantly different results for common bile duct and for gallbladder, and for both of two wavelengths excitation. And we obtained a different perfusion for these three different parts again. And our perspective study is about optical percutaneous needle biopsy, combined use of fluorescence spectroscopy and diffuse reflectance spectroscopy in one optical fiber. And again, we obtained a different fluorescence spectra for two wavelengths for excitation, and for diffuse reflectance spectroscopy for normal and malignant tissue. And our results, our modern fresh results for neurobiology is here. The study of adrenaline-induced monoamine oxidase dependent vasoconstriction in acute cortical brain slices. Again, we combine in one experimental setup two technology, fluorescence spectroscopy and video capillaroscopy. And we obtained results with a different reaction for adrenaline and siligilene. We proposed here the importance of monoamine oxidase and redox signaling in the mechanism of adrenaline-induced signal in astrocytes and cerebral vasoconstriction. And this is the identification of pathway regulation of a cerebral blood flow, and the assessment of pharmacological drugs as neuroprotectors is one of the ways of the cells protecting the brain from their death. And in conclusions, the obtained results demonstrate the ability of the combined OND techniques to prove useful information for diseases classification, for functional diagnostics, rheumatology, endocrinology, and so on in clinical practice, but the acute remains the search clearly expressed of a new diagnostic criteria. This is our team and our collaboration. And this is acknowledgements for patients and for funds. And thank you for your attention.